Hi all, Dr. Clark here again, and today we're going to start talking about how cells replicate. Um, we're going to start talking about mitosis, and that's somatic cell replication, and how do like normal skin cells, intestinal cells, muscle cells, how do these kind of cells, how do they replicate pretty much on a everyday basis, or at least a weekly basis? How are they replicating? Before we get into that, we're going to talk about a little bit about the differences between prokaryote and eukaryotic cell cycles. So here you can see over here, this is actually a species of trypanosoma. This is a eukaryotic cell, but it's going through cell replication much like a prokaryotic cell would. You can see where the nucleus is going to replicate itself, it's going to copy itself, and then it's going to divide other things that are in the cell, whether that would be like mitochondria, lysosomes, those kind of things. They're going to copy themselves. And then this is going to go through a similar function called binary fusion, where the two where the original one separates from the new one. Um, and that's kind of how prokaryotic cells break themselves apart. Now, remember, prokaryotes don't have membrane-bound organelles, so there's not going to be a nucleus in there. There's not going to be a mitochondria or lysosomes, whatever that's representing. But nonetheless, their DNA is going to copy and they're going to separate themselves. That happens along what we call the cell cycle. And so the cell cycle is going to have a couple phases. The G1 phase, which is the first growth phase. The S phase, which is the synthesis phase, where DNA is copied. The G2 phase, which is the second growth phase. And this is where things like mitotic spindles and things like that are forming. So it can enter the M phase. And the M phase is going to stand for mitosis if it's somatic cells, and if it's germline cells or reproductive cells, it's going to stand for meiosis. Okay. So that's kind of just an overview. Now we're going to go through and look at some specifics when it comes to prokaryotic cell cycle and eukaryotic cell cycle. Okay, so you know that prokaryotes are fairly simple, right? There's no organelles. They have a single chromosome. It's a circular chromosome. It's embedded into the nucleoid region of the prokaryote. But when it comes to complexity, we're just going to say that they're fairly simple. This works in the benefit for them because the more simple you are, the faster you can do reproduction. You don't have to wait to copy all your mitochondria, all your chloroplasts, all your organelles. You can just copy your DNA and split. And that's exactly what prokaryotes are going to do. So the first process is to copy that DNA. Now, in later lectures, we'll talk about the actual process by which DNA is copied. Right now, we're just going to call that synthesis, or DNA synthesis. It's replication of the DNA. We'll get into all the details about all the proteins and enzymes and whatnot to build a new strand of DNA. We'll get to that later. Right now, synthesis phase, or replication phase. And then, when it comes to prokaryotes, it's as simple as just splitting. We call it binary fission, but it's the process of just splitting the cell in two. Now, mind you, that both the new cell and the old cell, genetically speaking, they are identical because this is an identical copy of DNA. So this is a way that organisms clone themselves. And so prokaryotes often clone themselves. Some eukaryotes clone themselves. I mean, we clone ourselves every time we we cut ourselves or we make new intestinal lining and things like that. It's just a clone of the cell that's next to it. So we clone ourselves. But organisms like prokaryotes, single-celled organisms, will clone themselves for reproductive purposes also. Now, that doesn't mean that all bacteria are the exact same copy of each other. there's other ways at which genetic diversity can be incorporated into a bacteria. They can have things like plasmids, which are little teeny circular chunks of DNA that's specific to an individual. And they can have mutations that occur during some of these processes of replication that can make the individuals not identical. But through this general process of replication, binary fission, the individuals are identical, genetically speaking, at the end of that process. Okay. All right, so we talked about this before, but I'm just going to reiterate it. When we talk about prokaryotic chromosomes, whether we're talking about archaea, that domain, that kingdom, or bacteria, that domain, that kingdom, they have a single circular DNA, at least as far as we know right now. So their chromosome is not linear like our chromosomes. Their chromosomes are circular and they're connected, and they have one of them. The other thing that we'll come back to and talk about is their DNA is continuous in gene form, meaning that every spot on their circular chromosome codes for some type of protein. So it's continuous, it's back-to-back genes. Our DNA is not that way. And we talked about this a long time ago where I said there's 3 billion base pairs in the human genome, and we only have 25,000 genes. So roughly maybe 25 million, somewhere in that ballpark, of our base pairs code for something. Right? Code for some protein. About 25 million of our base pairs. The rest of the base pairs that occur in our genome are more like kind of placeholders. You can change them, but you can't delete them. We'll come back to this in a little bit. So, talking about prokaryotes though, single chromosome, circular. They have a region at which it starts to unravel, unzip. So in other words, the nucleotides that are together, the adenines, thymines, guanines, cytosines, the hydrogen bonds holding them together start to break. And they're broken by an enzyme called helicase. But that region where that starts to happen is called the origin of replication in prokaryotes. From that origin of replication, in both directions, new strands of DNA are being added. And so basically you're adding complements to the old strand. So we're going to watch a little video to see how this is done.
The genetic information in a bacterial cell is stored in the form of a double-stranded, covalently closed circle of DNA. Replication begins at a specific site called the origin. The origin replicates, and then DNA replication proceeds in two directions. The two original strands, shown as solid lines, serve as the templates for synthesis of new strands, shown as dotted lines. This is referred to as semi-conservative replication.
Okay, so you see there that even though it's circular and it's a closed system, you still get new strand bound to old strand, new strand bound to old strand. But these are identical copies of DNA. All right? So any cells that are produced from this, genetically speaking, they are identical. They're clones of each other. So once that replication occurs, so once the chromosomes have been replicated, for a small amount of time, the bacteria or the prokaryote is going to have two chunks of DNA, two chromosomes, until that bacteria can elongate and grow large enough that it would support the cytoplasm splitting in two. Right? So it has to reach a certain size. So at some times, it's quite possible to find bacteria and archaea that are in different stages of replication, and they would have two chromosomes. They would have two DNA molecules inside them. But that's, that's just the process by which they're waiting to grow. So there might be an environment where nutrition is not plentiful, so it's taking them a while to grow. Or you got lucky and you caught them in that stage. Nonetheless, eventually they'll get big enough and they can divide into equal halves. Now remember, when we were talking about bacteria before, they have at least 2 layers of protection, a plasma membrane and a cell wall. Now some bacteria will also have a capsule, a 3rd layer. So nonetheless, that material starts to stretch and be kind of pulled apart until it gets really thin, and then new layers will form around the DNA. And so you'll get the constriction of the cell to form 2 daughter cells. Now when we're talking about daughter cells, we're talking about identical copies, genetically identical copied cells. Alright, so let's look at this in graphical form. So again, origin of replication, DNA replicates, you got 2 chromosomes in there, the cell has to elongate, eventually new material starts to be laid down as the cell pinches in 2. Okay? It's a little bit different than when we talk about animal cells. Animal cells will form what we call a cleavage furrow, and they'll pinch in 2. Plant cells will form a cell plate where they don't get separated. But the other thing that can happen is sometimes there is no separation. Sometimes the bacteria don't pinch off and create 2 daughter cells. In other cases, there might just be what we call streptobacteria, and they might be in long chains, and they might be identical to each other because this one would replicate and then it'd be attached to the next one, on and on and on. So it doesn't always have to break free and have 2 separate daughter cells. Sometimes they remain connected through kind of a glycoprotein connection between the 2 to create these long chains of bacteria. And we'll look at these later when we talk about bacteria more. Alright, that's pretty much prokaryotes. Pretty simple process. Copy the DNA, stretch out, and break apart. Or stay together, but have 2 compartments in which your chromosomes exist in, or chromosome exist in. Eukaryotes much more complex. Again, for the most part, they contain more DNA. They also have a bunch of organelles. So for the most part, they're much more complex. Now, there are exceptions to every rule. Things like bulldog ants, and there's a few other invertebrates that have a range of chromosomes, and at times you can find certain species with a single chromosome. They're eukaryotes, but they only have 1 chromosome. And then you can have other cases where you might have an organism that you're like, well, that's not a very complex organism, something like a blue crab, okay, or a king crab. Most people would say, well, they're more complex than bacteria, but they have like 250 chromosomes, different chromosomes. So complexity is not directly related to how many chromosomes you have, and we'll get into this a little bit later. Remember, chromosomes are kind of the way the DNA is packaged. What the DNA codes for is directly related to the complexity of the organism, not necessarily how many chromosomes you have. But we'll come back to that. Okay, so nonetheless, eukaryotes, we've already talked about them. There's more DNA. There's more chromosomes. There's more organelles. Well, there are organelles. Okay? And so typically we would say that they're more complex. So the process is going to be more complex. Eukaryotic cells have linear chromosomes. We already talked about this. So it makes it for a little bit different in the way at which these chromosomes are copied. and separated. So there are two separate mechanisms for dividing up DNA. The first being mitosis, and that is the mechanism that would help divide non-reproductive cells. Now I want to make sure, even though I occasionally do this myself, and sometimes in the writing it looks like mitosis is cell division. Mitosis is not actual cell division. Mitosis is a mechanism for which cells can divide, and the mechanism that mitosis is, it is the division of the nucleus, or the division of the genetic material. So the genetic material is copied, and then it's divided through the process of mitosis, as long as we're talking about non-reproductive cells or somatic cells. Meiosis is a similar process of nuclear division. So again, this is not cell division. It's a process of cell division. It's a mechanism for cell division. Cell division is not just nuclear division. There's other pieces to it, and we'll get to that. Okay? But meiosis is the nuclear division of cells deemed sex cells or reproductive cells, and we call those germline cells. Okay, so two ways at which eukaryotic cells can divide up their DNA: mitosis and meiosis. Now, when eukaryotic cells divide, they divide through distinct phases. And most of you have probably heard some of the phases, but we're going to start with a phase that is now broken up. So when I was learning biology, we just called it interphase, and that referred to the first part of the cell cycle, or the main part of the cell cycle. Now, interphase has been broken up since into 3 different phases. So some people call them subphases, but these are actual phases, and interphase probably just needs to go away. The term needs to go away, because these phases we know a lot more about the G1 phase, the S phase, and the G2 phase than we've ever known. And they're legitimate phases, and we can tell when a cell is in each one of these phases. Overarchingly, though, we label them under interphase. Then there's the mitosis phase, or the M phase, if we're talking about somatic cells. If we're talking about germline cells, the M stands for meiosis. And then there is cytokinesis, or the C phase. Now, this is cell division. Right? This is a cell cycle. These two pieces are the most integral parts of cell division. Now, you could argue that it's not going to occur if you can't copy your DNA, so S has to be applied too. So in kind of overarching terms, this entire piece right here, interphase, mitosis, and cytokinesis, are the cell cycles, and when we talk about cellular division, they all have to have occurred. Mitosis is nuclear division, and cytokinesis is the division of the cytoplasm. So cytokinesis is the actual cell division. And the cell is actually splitting in cytokinesis. And we'll look at this all in much greater detail. So first, interphase. When I was learning general biology and I first learned about interphase, it was really considered the resting phase. We didn't know much about it whatsoever, and we just always would just consider, oh, that's the phase that cells are in doing their normal daily activities. Nothing interesting is going on in these phases. Well, we now know that that's not true. That interphase consists of the G1 phase, the S phase, and the G2 phase, and there's a lot of interesting things going on, including preventions of the other phases, so checkpoints. that occur in the interphases, and we'll get to that. So let's look at these three phases that make up interphase. So the G1 phase is the primary growth phase, and this is also the phase at which normal cell activity is occurring. So if we're talking about muscle cells, right, that are established, they're not reproducing, whatnot, they're just doing what muscle cells do, stretch, retract, these kind of things. G1 phase. We're talking about, nutrition cells, so cells that occur in your intestinal lining and your stomach. These cells, these microvilli cells, their job is to absorb nutrients, and that's what they do, and then dissolve it through enzymatic activity. Normal everyday activity, G1 phase. That's what the cells are sitting in. The S phase. The S phase is when those cells need to reproduce. Either they're damaged and they're going to reproduce a sister cell, or there's a cut, so epidermal cells, there's a cut and you need to heal the cut, that region. Just everyday activity also can cause these cells to need to multiply. So the cell would then move from a G1 phase to an S phase, and it would start replicating its DNA. After DNA replication, the cell would enter G2 phase, or sometimes called the second growth phase, and that's when the cell would ensure that the cytoplasm is big enough, that organelles have copied, that the meiotic spindle is formed or has the potential to form, and that the DNA has been copied correctly. We'll get to these checkpoints and we'll talk about them in a little bit, but that would be the G2 phase. From the G2 phase, the cells are going to enter the M phase. The M phase also has multiple phases. So mitosis has multiple phases, or what sometimes are called subphases, in which the cell would have different things going on inside the cell to help with nuclear division. The first part, or the general information that you need to know about M phase before we get into the subphases, is that the purpose of mitosis is to separate the copied DNA. It's separated by microtubule attachments and apparatuses that pulls chromosomes apart. So each sister cell would have the same number of chromosomes as long as we're talking about mitosis. Cytokinesis is then taking those sister cells and dividing them. So dividing the cytoplasm, dividing the plasma membrane, dividing the cell wall if we're talking about plants and fungi, that's cytokinesis. So the actual division of a cell is cytokinesis. Prior to that happening though, you have to have nuclear division, and that's mitosis or meiosis if we're dealing with sex cells. So here's an overview of the whole process. Again, interphase is just shown here as G1S and G2 phases just being combined. We'll go into more detail where I'll break this out and I'll show you that it's important that we really don't think of this as interphase, but we think of this as the G1 phase, the S phase, and the G2 phase. So what's going on? They're growing, they're copying chromosomes, they're copying mitochondria, they're growing cytoplasm, ensuring that there's enough nutrients that they can move into nuclear division. So that's step one. Step two is to enter prophase. That's the first phase of mitosis. Prophase is the only point at which you're going to be able to see chromosomes. Well, I shouldn't say that. It's the first point at which you're going to actually be able to see chromosomes. So if you find a cell in prophase, and I'll show you these when I demo out the mitosis meiosis lab, which is coming up soon. Okay, so you can look for that video. But you're going to be able to see the chromosomes. You can see them in prophase. Now you can see what looks like chromosomes in the end of G2 or in the G2 phase. But I'll tell you, you can't tell them apart from each other. In prophase, you can start telling them apart from each other. So the chromosomes are condensed. The nuclear envelope, so the membrane that holds the nucleus or covers the nucleus, it starts to dissolve. So those chromosomes can now float apart and they can move in the cytoplasm. And then these microtubule spindles start to form. Okay? And if it's an animal cell, it's coming from the centrioles. If it's a plant cell, it's coming from the Golgi apparatus. But nonetheless, these microtubules will form and they will eventually attach on to the chromosomes. So I want to make sure that you understand that these are not just like prophase and then straight to metaphase. Okay? So it's a continuous process. And so eventually these chromosomes are grabbed on by the microtubules. They grab onto the centromere, the center of the chromosome, and attach to them, and then line the chromosomes up in the middle of the cell, okay, on this imaginary plane, which is sometimes called the metaphase plate, sometimes called the central plane, the equatorial line. There's lots of names for it, but it's all imaginary, and that's just the center of the cell. And what's going on is these chromosomes are being jockeyed around and started to kind of be pulled loosely, because it's actually not being pulled, but the chromosome's actually eating the microtubule, and we'll get to this in a second. But those chromosomes are starting to move back and forth in the center of the cell, and eventually the centromere loses its force, the synapse between the two is lost, and the two sister chromatids are separated. So each side of the chromosome is separated, and they start to walk themselves away from the center of the cell towards the poles. And here you can see metaphase, they line up on the metaphase plate. Anaphase, those chromosomes, chromatids, are starting to move away from each other to the poles. Eventually, moving from anaphase to telophase, they get to the poles. A new nuclear envelope forms around them. The chromosomes can uncondense or uncoil, and then cytokinesis will occur, where if it's an animal cell, a cleavage row forms and they split. If it's a plant cell, a cell plate forms and they stay together. All right? So let's look at a video of how that works.
Most eukaryotic cells follow a process of growth and division called the cell cycle. These events include a growth stage, mitosis or nuclear division, and cytokinesis or division of the cytoplasm. Throughout interphase, the cell is engaged in growth and metabolic activities. Interphase can be further broken down into three discrete phases called G1, S, and G2. During the G1, or first growth phase, normal cell functions occur as well as cell growth. In the S phase, DNA replicates, producing 2 copies of each chromosome. During the G2 phase, the cell continues to prepare for mitosis and cell division. Mitosis, or the M phase, has 4 stages: prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes become visible and condense, becoming shorter and thicker. Each identical copy of a single chromosome is called a sister chromatid. The nuclear envelope breaks down and spindle fibers form as microtubules grow out of the centrioles that move to opposite poles of the cell. During metaphase, the double-stranded chromosomes line up along the equator of the cell. The spindle is now fully formed and the microtubules attach to each sister chromatid. Anaphase begins when the sister chromatids of each chromosome begin to separate. The centromere that holds sister chromatids together divides, and the chromosomes move away from each other along its spindle fiber. In telophase, the two groups of chromosomes reach the opposite ends of the cell. As a new nuclear envelope starts to form around each group, the chromosomes uncoil and the spindle disappears. The division of the cytoplasm and organelles is called cytokinesis, or the C phase. The result of mitosis and cytokinesis is the formation of two genetically identical cells. To summarize the entire cycle, G1, S, and G2 phases occur during interphase while the cell is growing and preparing to divide. Mitosis, or the division of the nucleus, consists of 4 phases: prophase, metaphase, anaphase, and telophase, and in these phases duplicate copies of the chromosomes are split equally. Finally, the cell's cytoplasm divides during cytokinesis and results in the formation of two genetically identical daughter cells.
Okay, so I hope that kind of gave you a better demonstration of how this works. We will come back especially to the prophase, metaphase, anaphase, and telophase section and the cytokinesis section in a future lecture, because I want to go into more in depth on what's going on, what's the actual formation, how do the centromeres get broken, how do chromosomes walk from the center, from the metaphase plate to the edge, to the poles, what's forming, what's going on, and we'll come back and look at that. We'll also spend more time looking at the interphase portion when we start talking about programmed cell death, cancer, checkpoints, these kind of things. So we'll come back and talk about those too. So this is kind of just an overview of the cell cycle. Now we're going to, for future lectures, we're going to dive into what does it mean to go through nuclear division, what does it mean to go through cellular division, and then how do we mitigate, or how do we determine what cells are going through cell division, what cells should not go through cell division, and how many times can cells divide. We'll come back and talk about those things. Till next time.